Good afternoon to all of you. And first of all, I thank the organizer for the opportunity to present my work and also to the chair's session. Okay, so as you see the title, effect of oxidative stress on biomolecule, any kind of stress in general is not good. Okay, so you can see in the left panel you have some oxygen radicals, and in the right panel you have some biomolecules, and I will be discussing the effect of these reactive oxygen species on some of the biomolecules. Mostly I will discuss about the proteins of two different kinds, elastin and lysozymes, and some real application of this ROS mechanism for two different strain of bacteria. One, I will take the example of E. coli. Already most of you have heard this term bacteria E. coli, and different strain of bacteria I will discuss about SORES. Okay. So one is gram negative, one is gram positive bacteria. So difference is their membrane content of the lipid in the membrane. And the motivation of this work comes from the optical tweezer experiment. So we face some challenges when we construct the molecule under the trap. So we have two bits. So if you consider two double trap, then you have two big microspheres trapped by the optical or the laser light, high intense laser light, and then you have some construct or anchoring molecule like DNA, and you have some molecule of interest. And when you ram the force, generally we faces the challenge that it often break the linker. Okay, so this breaking of the bond between this microsphere, which is coated by some protein and it is ligated with the DNA. So this is happening because of the oxygen radicals. Okay, so these oxygen radicals are damaging this bond, and that's why we got motivated to see the effect of these radicals on protein molecules. So the work I'm presenting here is mostly done by Devdeep and Rupal, and Akshay is in the audience, who has also presented a poster today. And Devdeep and Rupal is submitting thesis very soon. And I acknowledge the funding support from DST and most of the work in my lab and CRF, IIT KGP. So begin with, all of you know these are the oxygen radicals, and what it can do, so it can change the conformation of the protein molecule and it can degrade it. Okay. So this degradation is not limited only to the protein molecule, it can also damage to the DNA and lipid molecule. Okay. So this reaction oxygen species are key player in the growth of inflammation, cancer, or even in the neurodegenerative diseases like Parkinson or Alzheimer's. So those are the diseases caused by the intrinsically disordered protein, and that can be the conformation can be changed by this ROS. Okay, so common sources of oxidative stress, as I told you, you have some term like antioxidant or pro-oxidant. So in the body, it generates regularly, and there is a mechanism to neutralize these radicals. And if there is an imbalance of those radicals, then it can damage some of the protein molecule, and that leads to some kind of different conformational changes in the particular protein, and then it can later have some consequence of in terms of some diseases. So one example is here is the Fenton reaction, by which you can get the ROS. This term glycation, maybe it is not common to you, but this is related to the sugar. Okay, so glucose you intake, and that can also give you some kind of oxygen radicals, and it can change the conformation of the protein molecule or lipid molecule. Oxygen radical also can be generated by different level of exposure to the UV. So we can have UV radiation A, B and C depending on the wavelength, and some of the environmental stress will be there either through the cold or heat or any other external parameters. So I will be discussing about two different biomolecules, is a protein, elastin and lysozyme. I will not discuss this one, this will have a lot of data, so I will just skip that. So elastin is giving you the elasticity to the skin, okay, and if it is encountered some kind of radicals, oxygen radical basically, then it loses the elasticity. So we can estimate the changes in the conformation of the elastin molecule, how the content of their secondary structure changes by exposure to the oxygen radicals. Okay, so all of you have learned in 2.5 days about measuring the diffusion in various ways. So experimentally, there are many techniques by which you can measure the either translational diffusion or rotational diffusion. Okay, by tracking the particle, you can measure the RMSD and plot it with the T. Then a scaling parameter will tell you either it is following normal diffusion or ballistic or subdiffusion or superdiffusion. Experimentally, you can also do this very similar way through if the molecule size is very tiny, then very simple technique is laser light scattering, where you can have the ensemble average and time average are equal. You have a ergodic system, then you can measure the intensity correlation function with the time, and then with the help of Stokes-Einstein relation, you can measure the diffusion constant, and hence you can estimate the size of the particle in terms of hydrodynamic radius. So you can see here in the first bare elastin in 24 hours, the size is close to 350, and if it is encountered with the ROS, then the sizes drop to 250 or so. So the oxygen radicals are attacking or cleaving the sum of the secondary structure, or it is just destroying the sum of the bonds, mostly the SH bond. So you can see here it is growing like this and it is degrading. So maybe I will take 2 minutes. Okay. So you can see here it is following some kind of sigmoidal growth curve, which is very obvious, and when it is cleaving it, it is also following some kind of Z curve. So you can estimate the association or dissociation constant for this growth, and the change in secondary structure you can estimate through FTIR or CD. So basically some of the wave numbers are related to the secondary structure, just like here you can have for the beta sheet, or in this range for alpha helix, and another order structure or some turns in the protein molecules, and then you can have better estimate of each content by fitting this with the simple Gaussian functions. So you can estimate the content of the changes in the presence of some oxygen radicals. Okay. So there is another technique by which you can measure the secondary structure, just circular diachroism. So you have left-handed and right-handed Paul beams, and then absorbance depending on the wavelength, it can measure the secondary structure. So some models are there, you can compare that, and you can see the secondary structure of this one drop from 34% for the helical content, and even in the presence of UV in the C region, okay, so ultraviolet C, it dropped down to maybe 7% or so. So I will just go into the application part. So most of you heard about the superbugs or antimicrobial resistance. Okay, so you are consuming maybe antibiotic and it is not working on the bacteria, it is not killing it. So you have to have some alternative of that, and here some nanomaterial can serve the purpose, but the concern is it should be biocompatible. Okay, and that can be done by modifying the outer surface of the biomaterial, and here L-cysteine is a good choice, and it is having a kind of nanoflower structure, and if you just throw this into the some kind of bacterial cell culture, then it can kill the bacterial cell, and the killing efficiency is very high, close to 90% and above. So I can just skip and show you the final result. Okay, so this is the SEM image, and you can see here this is rod-like bacteria E. coli, and this is S. aureus, which is spherical in shape. And when you throw this kind of nanomaterial, then it can give some dent to this kind of bacteria, and the yellow curve is showing you the dent, and some the blue curve and the red curve is some of the cytoplasm is coming out of the bacterial cell wall. Okay. So with this, I end with the conclusion, and thank you for your listening.
Questions from the audience? It's a nice presentation. So I wonder, like these nanomaterials, you are saying you can use as an antibiotic.
Yeah.
So is it possible that in futures bacteria may develop also resistance to this kind of nanomaterial, or they cannot do this?
No, they, because see the bacteria has their own mechanism. Okay, so antibiotic, they can change their shape accordingly, they can modify, and then they are not working good in terms of killing those killing effect of the antibiotics, but these materials are externally given. So because you can also model the interaction of these material with the membrane. So wrapping and trapping, there is some kind of model where the interaction of this biomaterial with the lipid membrane, that interaction led to this kind of puncturing of the cell membrane, or it is going inside the cytoplasm and killing this kind of, because of this generation of ROS, there is killing those microorganisms.
Okay, let's thanks the speaker.
Thank you.